So we've been talking about how to organize animals in ways that make sense. So we talked about how at first we use hierarchical organization, but there was limitations to that because it didn't really tell you about the evolutionary relationships that exist between the animals, and then also didn't really tell you anything about the process of evolution and what led to that. And there's also differences in the similarities level of things which actually share equivalent levels of taxonomy. And so to solve those problems, scientists actually tend to use phylogeny instead of taxonomy nowadays, and we use that with basically these trees of life. But the biggest problems with the trees of life is that they don't really give you any information about how fast these animals are evolving. In other words, how long did it take place between that point and that point, Or, in other words, how long is this evolutionary process be taking place? It also doesn't tell you anything about how long have echinoderms existed? Have they been around since forever? Or are they kind of a new animals just because they're up here? Or has any animal been around for a very, very long time? They also don't tell anything about that point of origin thing. A lot of people get confused about whether or not the echinoderms and chordates come from each other because they're so similar. And so there's a lot of confusion by looking at trees of life. To solve these confusions, modern taxonomy and phylogeny and cladistics uses systematics and molecular biology to do something that we called a genome library and molecular clocking. So let's talk about that. When you look at an animal, you're looking at what the animal is actually expressing. In other words, the phenotype of the animal. But remember, the phenotypes are based on genotypes, or the genetic code of the animal. But just because you have a genotype, it doesn't mean you show it. Remember, there are silent traits which are not expressed, either because, for example, you are a diploid organism and you have two sets of genes. One gene is dominant over the other, so the other gene doesn't show even though you carry it. Also, some genes are masked by other genes in things like epistasis and multifactorial traits. There's also plate repeat to worry about there. And then you also have other things like silent mutations, which happen but don't show because they happen in the introns, or because they change the code without actually changing the message because of the ambiguity of the DNA code. We learned about that with the protein synthesis with the whole wobble thing. There's a lot of reasons why the genetic code might be different between two different animals, but they actually might still be similar in looks. Another thing that's interesting about the genetic code is that mutations happen throughout history with the code that you have, but you actually still have some of the leftover code from things that came before you. The same kind of way in evolution that we talked about vestigial structures. In other words, we have an appendix that we don't quite use anymore because it has some sort of vestigial function, or because our forefathers used to have it. In other words, why do we walk the way we walk, or why do we have the feet the way we have? Sometimes it's not because it evolved for the best, but because it is what our forefathers have. We just kind of change it for our own needs, but we are based on our forefathers. We are basically changed copies of what came before us. So you can't completely revamp a new organism. It is always based on the organism that came before it. So we have baggage. But just like we have morphological baggage, we also have DNA baggage. As the mutations happen, sometimes the leftover of what was before is still there, but mutated so much that it no longer has a functional purpose. We call it nonsense mutations. The DNA code is full, absolutely full of such junk DNA that has absolutely no function, but that it stores a history of our progression. So scientists can look at that, as well as fossil records and morphological similarities, vestigial, analogous, homologous structures, mosaic structures, to actually come up with a history of the evolutionary process throughout the history of life. And that's kind of like what we're going to talk about. So let's talk about the genome then as an evolutionary library. So there's two types of genes that happen in species. One is called paralogous genes, and the other one is called orthologous genes. Now, paralogous genes are basically genes which have been mutated from the original. So let's say, for example, the right way to do this is this way, but there's a mutate, or the original, the wild type is this way, but you mutated it. And now you have two different versions of that thing, that gene in the population. Now remember, we talked about evolution. If there's enough separation between these two things, it may have and enough subsequent mutations which cause more variation between the two species, eventually this could cause speciation. So paralogous genes or paralogous mutations will lead to speciation. But this gene 2 over here is paralogous to the gene 1. It's paralogous to the gene 1. I don't know how to say it, because the gene 1 is exactly the same thing as gene 2, but with a little bit of a difference, because gene 2 changed whatever was in gene 1. You follow what I'm saying? A good example of that would be something like, for example, the beaks of the finches are caused by several paralogous genes all getting together and causing a different speciation between the two species. But basically what happened there is that they have the original beak, which was changed because the genes for that original beak was changed. And so you may still have somewhere the original wild type there, but in environments where they were exposed to different things, mutations which occur that gave it an advantage actually made those become more common, and then a new kind of gene happened. So this kind of situation will lead to speciation, basically. All right? Now, if you actually have a situation where the gene gets duplicated in the genome, all right? In other words, you might not necessarily change the code, but you change it after you're already duplicated, then you might have what's called orthologous genes. Let me see what I mean by that. So the difference here is that in one case you have the original, and what happened is that you changed, you mutated the originals to maybe include something else in it that was not originally there. So this is a new version of a gene that was there before. So that will be a species, and that will be another one, because you actually changed the code. So that's paralogous genes. On the second example, what they're trying to show you is that you have an original gene, and then what happens is that instead of a mutation, like an insertion or deletion or something happening to change the gene, this happens to become duplicated in the genome. So you have a species that has that gene A, okay, which is in between those two marks that I did, twice. So it's still the same species, it's just having that gene twice. So that doesn't change the look, it just makes it double what you already had before. But that's not going to necessarily change your look, so you're still the same species. There's no speciation here because you didn't change the gene. But you have two copies of it. Now, what might end up happening afterwards is that maybe this copy there, the second copy that you had, all right, so let's say you had the original, and you have the gene A over here, but maybe the second copy of the gene A undergoes a mutation which changes it. Now you no longer have the original, you have a version of the original in here. But you still have the original there. You still have the original there. So that means you're not necessarily a different species yet, you're just a specimen that now has two kinds of that protein instead of one. A perfect example of this is what happened to the immunoglobulin gene with humans. We have one type of antibody, basically, that change into many types of antibodies because of series of duplications followed by mutations. Now, this could keep going, and then eventually you get a different variation with a C, and another one that's a D, and then maybe this B will actually become duplicated and form other variations of Bs, and so you eventually get a genome of the same species with hundreds of variations of that gene in it. And then maybe time goes by and a mutation happens on the gene A, which makes that non-functional, and then you don't have it anymore. But you might still have that stored in your genetic code as a record of the original one that you used to have. And so when you look at the genetic code, you can see these images of what used to be. And that is what we call a genomic library. For example, when you compare the code of two different things, you might see, like we talked about before in the lecture series, a degree of similarity between the genes. Notice here, they look different. But when you line them up with the computer software, you realize they're actually examples of paralogous genes. This gene just got some insertions that happened there. Or the first gene has it deleted, whichever one is the original, is what determines what kind of mutation happened there, right? Because 2 could have come first, and then 1 is a version of 2 deleted, or two came after, and that's because insertions took place. Whichever is the case, you know that that process led to the differentiation of the gene. And so, but you may still, if the orthologous process happened, have the original copy there, and then you have the new copy with the change, and then you know that what the original copy was, based on the library, basically. And you can use this to compare us to other animals. If you scan the code of humans, you will see there's actually a lot of orthologous genes which seem very similar to the genes that bacteria have. But we no longer use those genes. They're inactivated. They're mutated to the point that they're useless. But pieces of it are going to be homologous to the pieces that the bacterial genome has. And so we know that we share an ancestry with them because we have homologous DNA structures. Okay? And that actually explains why there's so much variety in life. Because you have paralogous genes, which are mutations causing adaptive radiation, and orthologous genes, which are mutations causing changes or more versions of the same kind of look. Together, you get a lot of variety of genes and therefore a lot of phenotypical variety. If there's a lot of genotypes, there's going to be a lot of phenotypes. And that's why there's so many different kinds of eye color. Because several genes for eye color have evolved. And so your eye color ends up being a combination of all of those orthologous genes. So this process helps us determine how to actually set up the history of the evolutionary process, because you can see, we can look at the DNA code as an actual evidence of what happened with it. Those deletions, those insertions, and whatever's left off can be used as an analysis to compare the DNA that we have with the DNA of other species to actually find out how close we are together. And again, the diversity that exists in the human population comes from a variation of both processes. Some people have genes which are mutated from the original or are paralogous to other people, and some people have different variations or different versions of the genes. They have orthologous copies, which creates more variation, or multifactorial traits, which leads to that. And now, on the next video, we're going to be talking about how molecular clocking is used to also trace the evolutionary process in terms of timing of the evolutionary process, based on the concepts that we talked about in this video. I'll see you guys then.